The function of neurons is to allow communication between cells, thereby maintaining homeostasis. Electrical signals, called membrane potentials, travel along the membranes of the neurons. Voltage variability and distance traveled determine the type of a nervous signal. Voltage variable short-distance signals are called graded potentials, while voltage constant long-distance signals are called action potentials. The distribution of charged ions close to neuron's membrane determines the membrane's electrical potential. The potential is measured in millivolts. A change in voltage results in the generation of a nervous signal. Two characteristics of cell membranes are needed to generate action potentials or membrane potentials and to maintain these potentials, and those are transport proteins in the membrane and resting membrane potential. Excitable cells need three different types of membrane ion transport proteins to produce and carry electrical signals. These proteins include pores, gated channels, and ion pumps. Sodium and potassium pores are always open, allowing these specific ions to diffuse down their concentration gradients. Like pores, gated channels are ion-specific. Gated channels are dependent on any of the three different stimuli applied to the membrane in order to open and close them. They're affected by certain chemicals, changes in voltage, or mechanical distortion. Chemicals like hormones and neurotransmitters bind to ligate gated channels to open and close them. These channels are typically located at the dendrites and cell bodies. Voltage-gated channels are used in generation and propagation of action potentials. They are located on the axons of neurons. Mechanically gated channels are opened or closed by vibrations or pressure stretching the shape of the membrane. These channels are located on the dendrites of sensory neurons or in the sensory receptor cells. The third type of membrane protein, the sodium-potassium pump, actively transports 3 sodium ions out of the cell for every 2 potassium ions pumped into the cell. This action helps maintain the ionic imbalances across the membrane as it offsets changes resulting from the ionic flows through the gated channels. This imbalance is known as the resting membrane potential. A neuron's membrane at rest is positively charged on the outside and negatively charged on the inside. A membrane with opposing charges is known as a polarized membrane. The difference in charge inside and outside the membrane only occurs close to membrane. The cytosol and extracellular fluids are electrically neutral. A typical value of the resting membrane potential is -70 millivolts. The resting membrane potential is maintained by an equal distribution of ions across the membrane. Second, a greater permeability of the membrane to potassium ions than sodium ions because of the presence of greater number of potassium pores. This results in a more positive ions leaving the cytosol and making the inside slightly more negative. And finally, the effect of sodium-potassium pump. Because some sodium is always leaking and potassium is leaking out, the pump helps the pumping out sodium as fast as it takes in potassium, thereby maintaining a difference in the gradients of these ions. The result is a constantly maintained electrical charge across the membrane. In summary, many potassium ions leak out of the cell, leaving the inside of the membrane slightly negative. A small amount of sodium ions leak into the cell. The sodium-potassium pump maintains the chemical gradients of sodium and potassium and provides a small change in voltage. The average voltage maintained across the resting membrane is about -70 millivolts. A variable deviation of the resting membrane potential voltage value is known as a graded potential. A graded potential occurs when the stimulus opens or closes gated ion channels and the voltage of the resting membrane potential is altered because of the changes in ion charge distribution. As the stimulus increases, more gates are affected and therefore the rate of ion flows as well. The greater the stimulus, the greater the change in potential, hence the name graded potential. These graded and local voltage deviations can increase the polarity, meaning more negative inside, or decrease in polarity, meaning less negative inside. An increase in polarity is known as hyperpolarization. A decrease of potassium ions inside the cell makes the cytosol relatively negative. A decrease in polarity is known as depolarization. An increase of sodium ions inside the cell makes the cytosol relatively positive. The role of graded potentials is to excite or inhibit action potentials. An action potential in the nervous impulse or signal for long-distance communication is established. Each action potential is generated at the cell's trigger zone. Generating and conducting an action potential requires both sodium and potassium voltage-gated channels. Through a sequence of electrical events, an action potential momentarily reverses the charge of the membrane and then restores the resting membrane potential. Before the action potential is generated, the membrane voltage is at the resting membrane potential. Sodium-gated channels must be activated to generate an action potential. Sodium channels have both activation and inactivation gates. The strength of the stimulus, graded potentials, determines whether the action potential will be generated. The voltage-gated sodium channels will not open if the graded potentials are hyperpolarization. Weak or subthreshold depolarizations will not open voltage-gated sodium channels. Only an above-threshold depolarization will open the voltage-gated sodium channels that generate an action potential. Depolarization of the membrane will continue until the charge or polarity is reversed. Because action potentials are either generated or not, they are considered all or none phenomena. The reversal of membrane charge begins the process of repolarization. Potassium channels come into play, allowing the potassium ions to flow out of the cytosol. Sodium gates close, repolarization continues, and the resting membrane potential is partially restored. Continuous redistribution of sodium-potassium ions by the sodium-potassium pump finishes repolarization and the restoration of the resting membrane potential. During the generation and conduction of an action potential, a refractory period exists. This means that the membrane cannot generate another action potential during this time.